Welcome back to Longevity Talk, and if you like this content, please consider subscribing to our channel. For centuries, humanity has searched for ways to slow the relentless march of time, to add not just years to life, but life to years. This quest has moved from myth and legend into the bright, sterile light of the modern laboratory. Today, we stand at a fascinating new threshold. Scientists have accomplished something remarkable, a feat that feels drawn from the pages of science fiction, but is grounded in meticulous research. They have successfully transferred a gene associated with a longer lifespan from one animal species to another, and in doing so, have extended the life of the recipient. This single act could ripple through the entire field of geroscience, the study of aging itself. This breakthrough did not emerge from a vacuum. To truly grasp the significance, we must understand the experiment's elegant design. Researchers focused on a well-studied gene, consistently linked to longer lifespan that supports cellular maintenance and stress resistance. It acts like a skilled manager in the cell, optimizes energy production, repairs damaged DNA better, and bolsters defenses against molecular damage. Scientists isolated this DNA segment from the donor species. Next came gene transfer, a core molecular biology technique. They needed a vehicle to deliver the gene, commonly a modified, harmless virus. Viruses naturally insert genetic material into host cells, and researchers harness that ability for therapy. They remove harmful viral parts and replace them with the longevity gene. The engineered virus becomes a microscopic delivery service, shuttling the gene into host cells. Animal choice matters. Typically well-understood lab species like mice, with short lifespans to observe lifetime effects, and clearly separated control and experimental groups. The results were striking, deeply encouraging for aging research. Weeks and months passed. A clear difference emerged. Controls aged as expected, with declines in activity, metabolic health, and cellular function. The experimental group was markedly different. They outlived controls, increasing average and maximum lifespan. They sustained a healthier, more youthful state longer, thicker fur, higher activity, stronger grip. Under the microscope, cells looked younger with fewer signs of molecular damage. Numbers confirmed it. Better energy management, more stable blood sugar, improved insulin sensitivity, lower inflammation. A single gene from another species orchestrated a coordinated anti-aging response. The gene-reading machinery proved compatible across species, suggesting fundamental mechanisms of aging are conserved across evolution, hinting these pathways could be targetable in humans, turning a compelling hypothesis into observable reality. The implications are far-reaching across science and medicine. First, it is a monumental proof of concept. A single gene moved between species can confer longevity benefits in a mammal. It shows the core programming of aging can be altered. It links pathways from simple organisms to complex ones. Second, it is a powerful tool for mapping aging biology. Does it enhance cellular repair? Optimize mitochondrial energy? Reduce senescent cells? Answers reveal critical network nodes, druggable targets. Conventional drugs could mimic gene effects. Third, while direct human gene therapy is distant, this opens nearer-term avenues. Understand the protein, then design a medicine to activate the pathway. A pill to bolster resilience to aging damage is more feasible to deliver. It signposts targets for health span-promoting pharmaceuticals. Ultimately, it helps shift our paradigm of aging, from inevitability to influence. It supports preventative medicine, targeting aging itself to delay diseases like heart failure and dementia. This experiment extends the boundaries of our medical imagination. Despite the excitement, scientific realism is essential. The biggest limitation is the biological gulf between small lab animals and humans. Humans are more complex, live longer, and have different metabolic and genetic networks. Many therapies succeed in animals but fail in human trials. Altering genes carries risk. Genetic systems are delicately balanced by evolution. Turning one gene up can have unintended consequences, including cancer risk. Exhaustive long-term safety studies across species are mandatory. Genes are dimmers, not switches. Dose and tissue control are complex technological challenges. Genetic modifications can be permanent. Precision is paramount. Aging is polygenic and shaped by environment and lifestyle. A multi-pronged strategy will be needed. This study is a vital clue. One piece of a larger puzzle. With powerful discovery comes responsibility to consider ethics. Equity and access. Who benefits if a therapy emerges? Avoid a biological divide between the long-lived and the short-lived. Policies must aim to ensure benefits reach all, prioritizing health span and quality of life. Next comes replication and expansion across labs and species. Larger mammals will help assess safety and efficacy. Mechanistic work will pave the way for drugs that mimic benefits without gene edits. Progress will be methodical under strict regulatory and ethical oversight. Years of preclinical work precede phased human trials, focused first on safety. Expect timelines measured in years and decades, not months. The message: balanced optimism. A key longevity gene can extend lifespan and health span across species. It is a fundamental proof of principle opening exciting avenues. Yet hurdles remain: safety, control, and human-animal differences. Science must proceed with rigor and strong ethics. Do not expect a fountain of youth soon. Avoid hype. We live in a remarkable era. This study is a milestone on the path to prevention. Today's proven tools: a balanced diet, regular exercise, sufficient sleep, and strong social connections. Stay curious, follow reputable sources and this journey of discovery.